So welcome everyone to this special edition of the Instruct-ERIC webinar seminar, Structure Meets Function. Today's is a special edition because we are featuring two colleagues from the University of São Paulo who has been doing very interesting work in structural biology by accessing research infrastructure available in this European network. So before starting, I'm going to do just a brief introduction of Instruct. Okay, so I hope you are seeing my presentation now. So Instruct-ERIC is the European Research Infrastructure Consortium for Structural Biology Research. It is the single point of access to technology and expertise for structural biology research in Europe. Instruct has more than 80 services available, among them sample preparation, biomolecular analysis, and 3D structural analysis. Instruct collaborates with institutions around the world, among them Instruct holds memoranda of understanding with 10 Latin American institutions, plus one facility in South Africa. Instruct has held 2 international calls in the context of the ULAC ResInfra project, so that researchers from facilities that hold most can apply to access European Instruct centers. So now there is a call open for monkeypox or SARS-CoV-2 structural biology research. So here you have a website where you will find more information about this. So with that, I'm going to finish this introduction. And okay, it is my pleasure to introduce our first speaker of today, who is Richard Garratt. Richard has been on the academic staff of the São Carlos Institute of Physics for more than 20 years. As a biological scientist by training, he is almost unique in Brazil for holding the position of full professor in the physics department. His postgraduate training was undertaken in crystallography and allied spectroscopic techniques at Birkbeck College in London during the 80s. He subsequently moved to the University of São Paulo, where he was instrumental in establishing the first protein crystallography laboratory in Brazil and possibly Latin America. He also actively participated in the proposal which led to the construction of the first protein crystallography synchrotron radiation beamline in the Southern Hemisphere. His interests span many aspects of structural biology, but principally the use of protein crystallography and homology modeling in understanding of a wide range of biological phenomena at the molecular level. Furthermore, his interest in the teaching of structural biology has led to the development of many very interesting pedagogical aids. His laboratory is the only Latin American participant within the international network of protein engineering centers, and he represents Brazil within 7, which is the Structural Biology Center of the Mercosur. He is a full member of the Brazilian Academy of Science and the Academy of Science of the state of São Paulo, and holds honorary doctorates from 2 Peruvian universities. So, Richard, the space is yours. It's a pleasure to have you here with us. Please.
Thank you very much, Natalia, for that kind introduction. I'm assuming that you can see my screen. Is that correct? Yeah?
Yes.
Okay, thank you very much indeed. So thank you also, Instruct, for the kind invitation to be with you today, and good morning or good afternoon to everyone wherever you happen to be. I apologize a bit for the rather cumbersome title of this talk, but I thought it was interesting, or an interesting idea, to include this question of research infrastructure in the title, given that this is an Instruct seminar, webinar. So as structural biologists, we are addicted to and dependent on research infrastructure, often large-scale research infrastructure, wherever it happens to be. But of course, in Latin America, it's not so easy to find research infrastructure on your doorstep. And that means we often have to travel abroad or send samples abroad, particularly more recently to Diamond in the UK for data collection. And SEBEM, which Natalia represents, and Instruct has recently been undertaking a landscaping activity where we have aimed to try and find out what research infrastructure exists in Latin America, how people use it, what people need, what are the bottlenecks, and what we can do moving forward to enable the Latin American structural biology community to be more competitive in the future. So hopefully this will lead at some point to a roadmap for future development of the field in the region. So this has been done under the auspices of the ResInfra EU LAC initiative, collaboration between Instruct-ERIC and SABEM, which is this community of structural biologists in Latin America. But what I'm actually supposed to be talking about are septins, which are filament-forming GTPases. And so all of the work I'll be talking about today involves data which has been collected somewhere around the world at one of the synchrotron sources, and more recently at the Brazilian synchrotron source Sirius, which is a 4th generation machine, recently come online, has one protein crystallography beamline, and here you're seeing an artist's impression of what it looks like. And housed at the synchrotron laboratory at CNPEM is also where we have the only cryo-electron microscope currently operational in Latin America. So research infrastructure is not as widely distributed as in other parts of the world, but we're trying to make as much use of it as possible. So what are septins? So septins are a rather neglected 4th filamentous component of the cytoskeleton. Most people are probably familiar with actin filaments, intermediary filaments, and microtubules. Septin filaments are narrower, they have a narrower diameter than all these other components, and they're probably less well studied. They're actually apolar filaments, and they're heterofilaments composed of several different septin molecules. And they're involved in a whole series of different important intracellular events, most of which involve, once the heterofilament has been formed, it will bundle together with other heterofilaments into higher order complexes, which will frequently associate with membranes and other components of the cytoskeleton in order to reorganize, restructure, deform, and remodel biological membranes. So they're involved in a whole series of events which involve these sorts of processes, going from cell division itself, exocytosis, vesicle trafficking. But they're also involved in forming barriers, for example, at the base of the primary cilium, in order to limit lateral diffusion within the biological membranes and to retain certain components within particular regions of the membrane. The most important point then about septins is they form heterofilaments, which bundle, interact with membranes, and interact with the cytoskeleton. But as I mentioned, they're GTPases. So a generic structure of a septin molecule involves a central GTP binding domain, which has something called a septin nuke element at the end of it. This 50 residue sequence characterizes a septin, distinguishes it from other small GTP binding proteins. And that's flanked on either side by an N-terminal domain, which is slightly disordered, and a C-terminal domain, which codes for coiled coils, which Italo will be talking about in the second talk this morning. At the end of the N-terminal domain is a polybasic region, rich in lysines and arginines, important for binding the plasma membrane. So if we're looking at human septins, they can be divided up into 4 different groups. There are 13 of them, and they get numbers, 1, 2, 3, 4, etc. But if you're hearing about septins for the first time, it's going to get very confusing if I start referring to them with their numbers. So since they can be divided into 4 groups based on domain structure and sequence identity, I'll use 4 different colors for the 4 different groups. So I'll have green septins, blues, reds, and there is only one yellow septin. And that will avoid the need to worry too much about the numbers. So as I say, they form heterofilaments. As I say, there's more than one septin involved in forming a filament. And in the case of humans, filaments can be formed either from a combination of 3 different septins, a yellow, a blue, and a red, or members from all 4 groups, including a green septin, so green, yellow, blue, and red. And these heterofilaments can be divided down into small oligomeric particles, which in the case of septins composed, or filaments composed of 3 different septins, are actually hexamers. So a hexamer is actually composed of 2 copies of a yellow septin, 2 of a blue and 2 of a red. And octamers, which are composed of 2 greens, 2 yellows, 2 blues, and 2 reds. So these are the fundamental particles which then polymerize in order to form a full filament. And there are many different combinations which are possible given all of these 13 different septins, but not any combination is possible. And they must obey a rule which we call Kinoshita's rule, which states that you must have the right colored combinations in order to form a full filament. You can't form a hexamer-based filament with a green, a blue, and a red, for example. It has to be yellow, blue, and red. So that limits the number of possible combinations down from nearly 2,000 to only 20 in the case of hexamer-based filaments and 60 in the case of octamer-based filaments. So if we look in the electron microscope with negative stain at low salt concentration, we see the filaments which have characteristic spaces between these filaments, and Italo will probably talk a bit about that shortly. But if we treat that at high salt concentration, what we see are these oligomeric particles. So in this particular case, it's a hexamer-based filament. So you can see 6 different blobs here in a row. And over the years, then we've accumulated a large amount of information about the crystal structures of the individual septins from the 4 different groups in the different colors here, and some other odd septins which don't fit nicely into this classification coming from other species. But this looks a little bit like stamp collecting, just solving more and more structures. But there's a lot of information, there's a richness of information in what I'm showing you here. But what we really need to do, obviously, is to look at the arrangement of the heteropolymer and not the individual septins. And the only crystal structure available, 2007, it was published by Wittinghofer's group, is one which is shown at the top here. So within the crystal structure, you see these filaments formed, and they're formed of the 3 different components, yellow, blue, and red in this case. And the filament extends throughout the entire crystal, and each monomer is stabilized on each side by 2 different types of interface. These interfaces are called NC and G interfaces. And there are 2 interesting points which come out of this crystal structure. First of all, what's the relationship between the trimer, yellow, blue, red, which is the content of the asymmetric unit, and the hexamer, which we see when we treat a filament at high salt concentration? The crystallographic symmetry gives rise to 2 possible hexamers, and I'll show you what they look like in a minute. And the second question which raises, if you look at the bottom where we're seeing crystal structures of individual septins, they all form basically the same sorts of filaments within the crystal structure. And when the interfaces which are formed between the individual subunits of these structures are not expected to appear in a heterofilament, we call those interfaces promiscuous interfaces. So for example, a blue septin would be expected to form an NC interface with a yellow septin and a G interface with a red septin. But when we crystallize it on its own, it forms a filament quite happily, forming promiscuous NC and G interface, which raises the question then of when all of these septins are being simultaneously expressed intracellularly, how does each septin know its rightful position along the filament? What are the details of the interfaces which guide the correct assembly? So these are the 3 possible arrangements for the hexamer. You could have yellow, blue, red, red, blue, yellow with a G interface exposed at the end with the yellow septin exposed in the G interface. Or you could have the one on the right, which has a red septin on the end and an NC interface exposed. Well, for more than 10 years or so, the community believed that it was this structure here on the left, which was the arrangement of the hexamer. We now know, we have just shown recently over the last few years that in fact that's incorrect. The hexamer actually has the structure shown on the right. And we did this different ways, and also Bill Trimble's group in Canada came to the same conclusion using different techniques. But I guess the final nail in the coffin was when we finally solved a cryo-EM structure for the hexamer at about 3.6 angstroms. And this clearly demonstrates that it is indeed the red septins, which are at the termini of the hexamer, exposing an NC interface. If you look at the chemistry of these interfaces, that makes a lot more sense, knowing that this interface at the end of the hexamer must be sensitive to high salt concentrations, because that's how we obtain hexamers from full filaments, by treating them with high salt concentration, high ionic strength. So we now know what the correct structure of the hexamer is. And the octamer is derived from the hexamer, not by adding extra septin monomers, the green ones, to the ends of the hexamer, but in fact they get inserted to the center of the hexamer. So the two green septins, which do not have the coiled-coil domain, which is the coiled-coil domain here, which Italo will be talking about, is represented by these stalks poking upwards, whereas the filament itself is stabilized by the GTP binding domain. And so the octamer is formed from the hexamer by inserting two extra subunits, the green subunits, between the two yellow subunits. And that means that you expose an NC interface in both cases. They can polymerize to form heterofilaments, which are composed of a mixture of hexamers and octamers. Okay, so the problem is neither the crystal structure nor our cryo-EM structure are high resolution, but it would be nice to be able to look at these interfaces in more detail. So one way of going about that is to divide and conquer. So by looking at the hexamer by looking at individual dimers within the hexamer, it should be possible to reassemble a full hexameric particle. So we have a crystal structure for a dimer of the two yellow septins with a G interface at the center. We have several structures for blue with red with a G interface between them. What we don't have is a crystal structure for the yellow with the blue, but we do have a promiscuous interface in the structure of yellow, such that we can use that to build a model for what would be the yellow-blue interface. And then we have everything which we need to reassemble a pseudo high-resolution structure for the hexamer. All of these crystal structures are of the order of two angstroms or better than 2 angstroms. And we can do exactly the same thing with the octamer because we have crystal structures for green with green, green with yellow. Once again, the weak link is yellow with blue, for which we don't have a high resolution crystal structure, but we can assemble all of this to have a pseudo high resolution structure for what an octamer should look like. So which of these interfaces do we best understand? Well, we have 3 different structures for blue with red. And if we look at them and map the interface residues, we see that there's a large interface and it's highly conserved. It doesn't matter which blue septin we pair up with the red septin, we basically get the same thing. And that would explain or provide a structural basis for this thing which we call Kinoshita's rule, which basically says that I can substitute a septin by another one anywhere along a filament, so long as it comes from the same group. So I can take the red septin, which in this structures is always septin 2. I can substitute with any blue one, 11, 8, or 6, and I basically get the same interface formed. But if I look at the other G interfaces which can form, in the case of hexamers, that's an interface between 2 yellows, and in octamers, it's an interface between a yellow and a green. It's a much smaller interface, which involves 2 particular, or it does not involve rather, 2 particular regions called switch 1 and switch 2, which in small GTPAs is normally used for signaling. So these are absent from the interface, whereas they're present in the interface with blue and red. And basically what that means is we have more and less stable interfaces. So the G interfaces between blue and red seem to be much more thermostable. That's what this histogram is showing. Whereas yellow-yellow in the case of the hexamer, or yellow-green in the case of an octamer, these are smaller interfaces, they're less thermostable. And that may be necessary in order for hexamers to transform into octamers or vice versa, because it's at the center where the 2 additional subunits must be introduced or must be lost. So can we explain, looking at all of this, why blue, for example, pairs up with red to form a G interface? And what we find when we look at these 3 different structures which we have is that there are subgroup-specific residues which are involved in forming that interface. So this particular histidine, for example, we take that as an example, it's only present in blue septins. And it forms part of an intimate interface with the red septin on the other side. No other colored septin would be able to form this interface because, as I say, the histidine residue is exclusive to the blue septins. Furthermore, it's connected to the gamma phosphate of the GTP via a network of interactions involving a threonine, which is also unique to the blue septin. So this network of interactions, which includes the gamma phosphate and leads all the way to the interface, is absolutely exclusive for the blue septins. And why is that interesting? Well, it turns out that the blue septins, like septin 6, are catalytically inactive. They're unable to hydrolyze GTP, whereas all the other groups do hydrolyze GTP. So this gamma phosphate will only be present in the blue septin structures. So we believe it's effectively part of the interface, which builds selectivity into this particular combination. But I just want to go quickly back to that interface, which we know less about, which is the one between blue and yellow. That's because this is an NC interface for which we have no high resolution crystal structure. So we have to go back to look at the cryo-EM structure. Now, what's particularly interesting about this interface is that is where the polybasic region resides. It forms a little alpha helix full of these basic residues here. And as I mentioned at the beginning, the polybasic region is fundamental for binding to the membrane. However, in this structure, we see that all of these basic residues are pointing inwards towards the interior of the structure and not exposed such that they could bind to a membrane. Well, all of the interfaces that we look in, all of these crystal structures, they're basically highly conserved with the exception of one interface, and that is this NC interface formed between two green septins, which be at the center of an octamer. And what we observe there is when the structure is bound to GTP, this interface is closed, and when it's bound to GDP, it's open. And what that means is this potentially couples GTP hydrolysis by the GTP domain to the opening and closing of the NC interface. That requires information to be transmitted from a G interface to an NC interface because the nucleotides are bound at the G interface, but the movement occurs at the NC interface. So what happens then is as that interface opens and closes, a whole bunch of salt bridges, which is the reason why these interfaces are sensitive to salt concentration in the first place, a whole bunch of salt bridges get completely rearranged. Very difficult to predict based on looking at the sequences, but this could possibly occur in two different ways. There's two different solutions to forming the salt bridges, which allows the interface to open and to close. And why is that important? Well, when the interface closes, that alpha helix, which has the polybasic region, there is no longer space within the interface for that little alpha helix to reside. So it inevitably has to get pushed out of the interface. You could predict that just by looking at the structures, but we actually have one structure where the alpha helix is present and we see it out of the interface. So what that seems to suggest is that the hydrolysis of GTP could render a mechanism by which the neighboring NC interface opens and closes. And as it does so, it either exposes or buries this little alpha helix, which has the positive charges on it. Once exposed, those that polybasic region will be able to interact with negatively charged lipids in the membrane, which are the type of lipids which we know they interact with. So what we have here is a mechanism which probably relates GTP hydrolysis, not to the formation of the filament itself, but more to how the filament interacts with membranes, how it binds and releases from membranes as a function of GTP hydrolysis. So if we just quickly go back to looking at what a hexamer looks like and an octamer looks like, the difference is having the two green septins inserted into the center of the particle. So what that means is that in any filament, you'll have stoichiometric quantities of red, blue, and yellow, but the green septin will be present in the dose which depends on how many octamers you have on your filament. So you could go all the way from a filament which just has hexamers to one which just has octamers to anything in between, because the interfaces at the ends of these two particles are common and they're able to couple. Since we have observed that the interfaces between green-green and green-yellow are weaker, they're less thermostable, they're smaller, and that's presumably related to the fact that the green septin can fall out of an octamer to form hexamers. That's often what occurs when we try and purify octamers. We often find we have many hexamers as a result. So this lability at the center of the octameric particle is probably important. And what that means is, given that it is the interface which can open and close as a function of GTP binding and hydrolysis, this means that different filaments which have different doses of octamers within them probably have different properties. Their ability to associate with membranes rather than components of the cytoskeleton could vary as a function of how much octamer, therefore how much green septin you have within your filament. So this gives us a wide range of different filaments going from the extremes of just hexamers to just octamers. And so that's kind of our current view of what the filament looks like. Italo will be talking in a minute about the coiled-coil domains. But basically, summarizing what we've learned so far is that everything really depends on what's going on at the interface. So specific interactions at particular interfaces give rise to the correct order of the oligomers such that they can spontaneously form. There can be opening and closing at the central interface, allowing the octamer to expand and shrink. Communication between interfaces, because where the GTP is hydrolyzed is not the interface where the change occurs. So there must be transmission of information from two neighboring interfaces. And well, something I haven't talked about, which is strand slippage, which occurs in some of the structures involving the interface of the G interface itself. So once again, thank you very much for your attention. Thank you for the invitation. And if there are any questions, I'd be happy to try and answer.
Thank you so much, Richard, for a beautiful talk. So now we are open to questions. You can write them down in the Q&A. So for the moment, I don't see questions in the Q&A. I don't know, maybe, or in the chat. No. Okay, so I have a question for you, Richard. I've heard in different situations, in congresses, or that the student has asked you, why did you move to Brazil when you were young? And do you hear me okay?
Yes, I can hear you, yes.
Yeah, but I am more curious about why did you decide to stay in Brazil? And in doing so, how did you manage to lead all this research, considering the infrastructure that you had available at different times in Brazil?
Right. Oh, that's quite a big question. Many different aspects to it. I think one of the reasons I decided to stay is because I could see that this was a land of opportunity, in a sense. If I'd stayed in the UK, I would be one of many, many protein crystallographers. When I arrived in Latin America in 1989 with Glaucius Oliva, we were the only protein crystallographers basically on the subcontinent. And that has opened up many doors. That's made for my career probably not to be quite as productive as it might have been if I stayed in Europe or gone to the US, but it's certainly made it very interesting. So I've had the opportunity to establish collaborations and run workshops and establish friendships all throughout South America. So I think it's one of those choices you have to make in life. What sort of career are you interested in? It wasn't a conscious decision. It wasn't what I had in mind when I came to Brazil, but it's probably what has led to me staying here. I think there are opportunities here. It's tough. We've been talking about research infrastructure or lack of research infrastructure. I'm in a very lucky part of South America. We do have a synchrotron only about 2 hours drive away. So I'm in the state of Sao Paulo, which is very well funded. So our laboratory is reasonably well equipped to the point where we are able to do research which may not be quite at exactly the very top level that we would like to be at, but that's where we're always trying to get. So I think one of the things which has helped is the fact that obviously opportunity may not be equally distributed, but intelligence and people's capacity to work is equally distributed. So we've been very lucky in attracting very good students who have all sort of embarked on this initiative to try and do something in structural biology in Latin America, and I've viewed it in the same way that I do. It's an opportunity, it's a challenge, but that can make it exciting. And I think that's probably the best answer I can give you right now.
Thank you so much. Now we have two questions in the Q&A. Do the octamer recognizes membrane curvature or it induces it?
Do the octamer recognize membrane? Oh, that's a very good question. Yeah, I took a slide out about membrane curvature. This is a kind of a hot topic. Septins recognize the curvature of membranes, specific curvatures. At least they're said so to do. But whether they induce curvature or just recognize it is still an issue which we're not 100% certain about. At least I'm not. I think the jury's still out on that, although there are many people who believe that that this is well established. And how octamers and hexamers may behave differently in this respect is also an excellent question. One would expect, due to the weakness of the interfaces at the center of the octamer, and the fact that the green septins lack the coiled coil, which Italo is about to talk about, probably gives them more malleability, more flexibility at that point. So it may well be that octamers are more able to form curved structures more so than hexamers. And so then what that means is the dosage of octamers in your filament may give it different properties with respect to its capacity to recognize membrane curvature. That's very interesting question. And okay, so what's the real function of GTP? Well, we're speculating, hi Rick, thanks for the question. We're speculating that GTP binding or hydrolysis could control the way in which the filament binds to and is released from membranes. This must occur if we stop and think a little bit about the physiological processes that septins are involved in, because the rings must shrink, for example, during cell division. So probably this is a process which involves binding to membranes and then being dissociation from membranes. And this may be related to GTP hydrolysis. But there's still lots of work to be done there.
So we have 3 more questions. Maybe we can take, maybe Richard, you can choose one of them and then answer the others.
Take the first one from Francois. Thank you very much for the question. Have any idea of the septin further interact with? Oh, okay, yes. Well, it's well known that septin filaments do interact with other components of the cytoskeleton. So they interact with actin filaments and they interact with microtubules. The details of these interactions are still very poorly understood, but septins are not the motive force involved in cell division, for example. So that involves the actin-myosin skeleton. So exactly how septins relate to and are involved in other processes which depend on actin, for example, once again, are still being investigated. But yes, they do definitely interact with other filamentous components of the cytoskeleton.
Okay, thank you so much, Richard. I was, if you agree, maybe you can answer the other questions just by writing to the people. Okay. So if they stick to our schedule, okay. And now we can move. Thank you so much to everyone for writing down their questions. And now we can move to our next speaker, who is Italo Cavini. Italo obtained his bachelor in physical and biochemical sciences from the University of São Paulo, receiving the Bernhard Gross Prize for the best academic performance among the cohort of students in that year. Then he obtained a PhD in biomolecular physics in the same university, working on the beta amyloid peptide studied by high pressure NMR with a sandwich period at the Universitat de Regensburg, Germany. Since 2019, he is a in 2019, he is a postdoctoral researcher at the University of São Paulo in the Physics Institute of São Paulo, working on the structural characterization of septin coiled coils. Currently, he is a visitor postdoctoral researcher at the School of Chemistry University in the University of Bristol in UK. So, Italo, it's a pleasure to have you here today with us. The space is yours. Please go ahead.
Thank you, Professor Natalia. Hello, everybody. So my name is Italo. Today I'm going to talk a little bit about the versatile septin coiled-coil domains, which is the work that we do in University of São Paulo in São Carlos, Brazil. But at the moment, I'm talking from the UK, where I am a visiting researcher here in Bristol. So as Richard presented, septins are proteins involved in the many cellular events in eukaryotes, mainly in membrane remodeling and scaffolding. And the septins have three main domains: the N domain, the G central domain, and the N-terminal domain, which contains the coiled coils I'm going to talk about. In humans, we have 13 septins. We divide them in 4 groups, and I'm going mostly to talk about the blue group, which is the septin 6 group, together with septin 8, 10, 11, and 14, and also the red group, which is the septin 1, 2, 4, and 5. So the polymerization model for the septins that we have currently is like it's shown in this picture. It's a hexameric or octameric particle. And in each position, you have a member of one of those groups that I mentioned. And it can polymerize by end-to-end associations through this NC interface. And once it's polymerized, we can recognize the presence of 2 different types of coiled coils. One, it's a homothermeric coiled coil between septin 2 members, which is a shorter coiled coil, around 30 residues. And we have a second one, which is a heterodimer between septin 6 members and septin 7. Which is around between bigger than 60 residues. But unfortunately, they are not visible in all the G domain septin structures solved so far. We have FRET experiments in the literature indicating that both of them are parallel. And we also have biophysical data suggesting that septin 6 and septin 7 C-terminal domains, when mixed together, form heterodimers. Okay, so here we have in this box representations the central coiled coil part of the red group, and also for the blue and yellow group. At the very end of these long C-terminal domains, we have a polybasic region, which I'm going to talk about at the end of my talk. But also coiled coils has been involved in coiled coils have been involved in interfilament bridges. So we have in yeast septins, which are the CDCs. We can see in the microscopy studies this kind of railroad tracks with density in between, which people believe that it's from the coiled coils. And more recently in mammalian septins, also this idea came to light. All right, so just talking a little bit about what coiled coils are. They are formed based on heptade repeats, in which the first and the fourth position are usually occupied by hydrophobic residues, which I'm going to indicate by this magenta color throughout my talk. And also they are characterized by the knobs into holes packing. So which is a very specific packing in which a side chain of one residue can be inserted in a hole formed by 4 other residues from another helix. And because this stripe is not totally vertical, the helices have to deform, so it creates a left-handed supercoiling twist, characteristic of heptads. But if we have, on the other hand, different periodicities, like hendecads or pentadecads, this twist changes, going from totally left-handed to right-handed. So it's there's a link between the twist and the periodicities you have in a coiled-coil. In this case, I'm showing for tetramers, but the same applies for dimers as well. All right, so I'm going to start talking about our results. First of all, about the structural characterization of these domains. And starting with septin 5, we solved the structure of this peptide together with a postdoc in our group, Diego. And it's what we say canonical parallel coiled-coil with hydrophobic in positions A and D. But on the then we solved structures from 2 other members of this red group, septin 1 and 4. And by our surprise, it was the orientation was anti-parallel. And this structure has 2 sides because it's anti-parallel. It has a hydrophobic side composed by the D positions and has a hydrophilic side composed of A positions. And interesting enough, this A side forms a hydrogen bond chain throughout the structure. So indicating that even it's not totally hydrophobic, it can contribute to the stability. And the same happened, this I'm showing for septin 4, but the same happened for septin 1, which is a very similar structure. All right, then we are interested about what the behavior would be in solution. So I acquired NMR spectra in Universität Regensburg together with Claudia and Hans Holbert. And we analyzed 2D nosy cross peaks and compared them with modeled spectra from anti-parallel and parallel models. And by analyzing this INOEs, we could find that septin 2 is anti-parallel, as can clearly be seen in these contacts that I'm showing as an example. But no anti-parallel contacts were found for the other 3 peptides. So they're probably parallel in solution. Okay, then I switch to the other coiled coils, the longer ones, from the blue group. And we solved also homodimers from this group, and they're also anti-parallel. And in this case, we have kind of an insertion in the center here. It's a stutter, but you can also represent it by a handicad. So it changes the twist, so it's not as twisted as a canonical coiled-coil, but it has 2 sides as well as the same that I showed you before for the red group. One side is totally or mostly hydrophobic, and the other side is hydrophilic, but not only hydrophilic, it has glutamic acids and lysines, and they're put in a favored intercalated way, so it can provide also stability for the coiled-coil. Fernanda, which was a former PhD student, also solved the structure for the entire C-terminal domain of septin 8. And they're very similar structures, but it was a nice proof that the truncation that we did for septin 6 did not change the result of the structural characterization. All right, but this kind of antiparallel coiled coils are not expected in the canonical model. Which is expected is the parallel heterodimer, which we also recently got a structure. We had many data sets combining different members of the blue and septin 7. But interestingly enough, septin 14 and 7 has the most, it's the most thermally stable. So I believe that's why we were able to solve this structure. And it's also very puzzling because it contains two different coiled-coil handedness. It starts with canonical heptads, hendecads, and then at the C-terminus, it's left-handed heptad-based coiled-coil. So, and this is nicely seen in the periodicity plot and also in the supercoiling angle. All right, and why we believe that there are different periodicities within these coiled coils? So my answer would be that it's probably very important for the mechanism of septin recognition. So for example, if you have a septin 6 and septin 7, it can form a coiled coil because it matches the periodicities. But on the other hand, if you have a septin 2, for example, which is very closely related to septin 7, it cannot form because it has a periodicity mismatch. And also we can extract insights from why to answer why the heterodimer is more stable in solution. So we believe that the heterodimer is longer. It could be around up to 90 residues in comparison to the antiparallel septin 6 coiled coil. So that's why in solution we mostly have heterodimers and not homodimers of 7 or 6. But these are probably in cell regulated or triggered by an event that helps triggers this conformational exchange, conformational change. All right. I also was lucky to be in a week in CHERM, which is an MR center in Italy, and it was also it's provided by through Instruct, and in this week I tried to do many things. So we did mutants of these shorter peptides, which at the end, in order to change the orientation we saw, but at the end we solved one of these mutant structures, and it was very, very similar, pretty much the same of the wild type. And I also did some preliminary tests on septin, on the longer one, septin 6 and 7, which now I'm continuing here in Bristol. And yeah, so I was really grateful to meet Professor Simoni and Massimo there. So it was a really nice experience. And to wrap this part, so we believe that these coiled coils have an intrinsic orientation ambiguity. They can have the parallel orientation in a filament, but when there are a lot of filaments in the neighborhood, it can create this kind of railroad tracks connected by anti-parallel dimers between septin 6 and septin 7 maybe, but also form this kind of sheets of filaments, and mostly connected by septin 2 coiled coils, anti-parallel coiled coil. And we believe that the lower dielectric constant near the membrane could enhance this polar interaction seen in the A side of the hydrophilic residues. Okay, then I'll move on to another part of my project, which is in collaboration with other students, which is interaction with other partners, non-septin partners. And this was done in collaboration with Daniele, which is a former PhD student as well. And we already know that Borgs or CDC42 EPs interact with septin through this heterodimeric coiled coils, septin 6-7. The literature has also shown that mutants of this GPS motif and LXX VL motif to alanine eliminates interaction. However, structural details are still missing. So we took a look at that. Here are some CD thermal denaturation showing that when we use the BD3 domain of the Borg together with septin 6 and 7, we see an increase in TM, but not when we use the mutated peptide, the control peptide, both for the C-terminal domain and also for a truncated construct. And by the emergence of AlphaFold, we built a trimeric high confidence model of this interaction. And interestingly enough, the BD3 binds to the handicap portions that I mentioned before. So it's in the N-terminal part of this these two chains, and it binds to the groove of this septin C-termini. And based on the model we built, we designed mutants to show that some residues are important for this interaction. So here are the same, here are CD thermal denaturations using a mutant of septin 7, lysine to arginine. It decreases the gain in TM, and this one totally eliminates interaction, showing that the model is probably the one that I showed you before, or it's very similar to the one that we see by AlphaFold. All right, then to finish this, I'm going to present a little bit about what I've done with interaction with lipids. It's just preliminary data, and these are what I call the polybasic regions in the C-termini. It's in the very end of the septin 6-like septins and also septin 7 throughout different organisms. So it's like a conserved feature. And this hasn't been explored much. So people don't know what it does, but it's probably doing something important because it's conserved. And so what I did together with former master's student Marina and Professor Ana Paula, we did some assays called PIP strips. So we have a membrane with some lipids in like this kind of spots, and we incubate the protein and we reveal using specific antibodies. So in this case here in the left, we have the full C-terminal domain of schistosoma mansoni septin 10. and it lights up some of these spots, mostly the ones from PIP2 lipids. But when we eliminate this polybasic region, it almost eliminates also the interaction with the lipids. But there's still the same pattern present, although less eminent. But when we use the full G and C domains, not the N terminal, but the G and C domains of septin 10, it also lights up more or less the same spots. But when we remove the 43 final residues, it totally eliminates the interaction. So it might be that this region is crucial to interact with lipids, but we don't know how it interacts with in coiled-coil or with just helices. So we're still trying to figure out what's going on. So as take-home messages, I expect that I showed a little bit about the septin coiled-coils and how they are metastable and multifunctional. I always like to have this scale in my mind. So we have coiled-coils which are more or less like scaples. So they're hyperstable, monofunctional, and most of the times very well comprehended. But on the other hand, the septin ones fall in a different category, like Swiss knives. They're metastable. They can function in different ways, and they're due to this very puzzling. And so the parallel coiled-coils probably stabilizes the NC interface in a filament, whereas the anti-parallel works as interfilament bridges. And this can help forming bigger and high scale bundles and sheets. Coiled-coil periodicities could play an important role and in how the septins can find each other. And as an ultimate message is nature trying to show us how to do more with less. So just by using one gene, you can have all this kind of interactions and structures as well. So just to thank my group in Brazil, and also Humberto, Professor Natalia, and Richard. Also collaborators from Sweden, Germany, Spain, and also here in UK, and also Professor Wolfson and Crump, which are my hosts, which are hosting me here in Bristol, and Fapepi for the financial side, and Instruct. We had 3 different projects approved, which are grateful for that, for accessing different infrastructures, and also pretty much in Diamond, analyzing our crystals, but also in Alba in Spain, University of Regensburg, and also here in Bristol. So thank you, everybody.
Thank you so much, Italo, for a great talk. So now we are open to questions that you can write down in the Q&A. I think there is a delay. Okay. I don't see questions yet. Richard,
No.
yeah.
I'd just like to thank
okay.
Italo.
Sorry.
Just like to thank Italo for his presentation, and particularly for showing the last slide. For some reason, I forgot to show my last slide with all the acknowledgements. So I'd just like to thank Italo for acknowledging all of the people who should have been acknowledged at the end of my talk also. And I apologize for my failure in that respect.
Okay, great. Thank you. Yes, okay, we have a question. Is it possible to assemble 7 homodimeric coiled-coils as done by the SEP6 group members?
Yes, septin 7 in vitro is dimeric as well when it's not put together with 6. And AlphaFold models show that it also can form antiparallel homodimers. So we believe that the same thing that happens with 6 could happen with 7. Yes.
No open questions. Well, I have a question. In general terms, how good do you think is AlphaFold to predict the interfaces?
Yeah, to be honest, I was kind of surprised by how good it can be.
Okay.
And now with Multimer, which is an evolution of the original AlphaFold, we have been getting nice results like tight complexes, and yeah, so it can it's really astonishing what it can achieve. Yeah.
Okay, good news. Okay, I don't see other questions in the Q&A, and I'm curious, more general questions. If you have any thought or comment to share with, let's say, young students starting a career in structural biology in Latin America.
How is it?
Do you have a thought, or if you have a thought or something that you would like to share?
All right. I think I'm being blessed with all the collaborators we have. So in Brazil as well. So I've been measuring in São José do Rio Preto and also in São Paulo, the University of São Paulo Institute of Chemistry. So I think establishing connections is really important, and also with Instruct. So we got the transition between the old synchrotron in Campinas to Sirius, and then we got like in a window that we could not send crystals to, and it was nice to have Instruct providing beamline time in Diamond that it was when one of these structures were solved. So yeah, I would say that it's important to have collaborators, and yeah, mostly that.
Okay, that's great. So I'm going to thank you again, Richard and Italo, for being here today with us. Your talk has been super interesting. Okay, so I think announcements will be of the next series of this webinar, of next session of this webinar series, and it's been a pleasure, and I think we can say goodbye for now.